Welcome back to a series of lectures. Today we are talking about the spontaneous generation. That's part 2 of the history of microbiology. The lecture is presented by Microbiology Biological Sciences. Kindly subscribe to our website to stay tuned. The spontaneous generation. In the second half of the 19th century, many scientists and philosophers believed that some forms of life could arise spontaneously from non-living matter. They call this hypothetical process spontaneous generation. About 100 years ago, people believed that toads, snakes, and mice could be born of moist soil, that flies could emerge from manure, and that maggots, larvae of flies, could arise from decaying and corpses. Francisco Redi's experiments. Physician Francisco Redi set out in 1668 to demonstrate that maggots did not arise spontaneously. Redi filled 2 jars with decaying meat. The first was left unsealed, allowing flies to lay eggs on the meat, which developed into larvae. The second jar was sealed, and because the flies could not get inside, no maggots appeared. Still, Redi's antagonists were not convinced. They claimed that fresh air was needed for spontaneous generation. So Redi set up a second experiment in which he covered a jar with a fine wet instead of sealed it. No larvae appeared in the gauze-covered jar, even though air was present. Redi's results were a serious blow to the long-held belief that large forms of life could arise from non-life. However, many microbiologists still believe that small organisms were simple enough to germinate from non-living materials. The case for spontaneous generation of microorganisms seemed to be strengthened in 1745 when John Needham found that even after he heated chicken broth and cooked broth before pouring them into covered flasks, the cooled solutions were soon teeming with microorganisms. Needham claimed that microorganisms developed spontaneously from the fluids. 20 years later, Lazzaro Spallanzani suggested that microorganisms from the air probably entered Needham's solution after they were boiled. Spallanzani showed that natural fluids heated after being filled in a flask did not develop microbial growth. Needham responded by claiming that vital forces necessary for spontaneous germination had been destroyed by the heat and was kept out of the flask by seals. Spallanzani's observation was also criticized on the ground that there was not enough oxygen in the sealed flask to support microbial life. Theory of biogenesis. In 1858, Rudolf Virchow challenged the case for spontaneous generation with the concept of biogenesis, hypothesized that living cells arise only from pre-existing living cells. Because he could offer no scientific proof, argument about spontaneous generation continued until 1861, when the issue was finally resolved by the French scientist Louis Pasteur. Pasteur demonstrated that microorganisms are present in the air and can demonstrate and contaminate sterile solutions, but that air itself does not create microorganisms. In 1861, he filled several short-necked flasks with beef broth and then boiled the contents. Some were then left open and allowed to cool. In a few days, these flasks were found to be contaminated with microorganisms. The other flasks sealed after boiling were free of microorganisms. From this result, Pasteur reasoned that microorganisms in the air were the agents responsible for contaminating non-living matter. In 1864, Pasteur next placed broth in open-ended long-necked flasks and bent the necks into S-shaped cups. The contents of these flasks were then boiled and cooled. The broth in the flask did not decay and showed no sign of life even after months. Pasteur's unique design allowed air to pass into the flask, but the curved neck trapped any airborne microorganisms that might contaminate the broth. The following are the pictorial presentation of these Pasteur's presentation and the experiments that he did to illustrate that microorganisms could not originate spontaneously. Pasteur showed that microorganisms can be present in non-living matter, on solids, in liquids and air. He also demonstrated that microbial life can be destroyed by heat and that methods can be devised to block the access of airborne microorganisms to nutrient-rich environments. These discoveries form the basis of aseptic techniques, procedures that prevent contamination of unwanted microorganisms, which are now the standard practice in laboratory and many medical procedures. And the aseptic techniques are very key in microbiology. Pasteur's work provided evidence that microorganisms cannot originate from mystical forces present in non-living materials. Rather, any appearance of spontaneous life in non-living solutions can be attributed to microorganisms that were already present in the air and in the fluids themselves. Scientists now believe that a form of spontaneous generation probably did occur on the primitive Earth when life first began, but they agree that this does not happen under today's environmental conditions. Thank you very much for viewing our lecture that was on spontaneous generation. Next, we'll talk about the first golden age of microbiology. Kindly subscribe to our channel to view more of such tutorials as they unfold. The lecture is presented by Microbiology Biological Sciences. Thank you and stay tuned for more lectures as they unfold. Goodbye.